Hello students, today we discuss about our chapter like the molecular basis of inheritance. In this chapter we deal the study of DNA replication. This DNA replication is having two types, that is heterocatalytic function and autocatalytic function. This DNA having two types of function, that is heterocatalytic function and autocatalytic function. Then what is meant by catalytic function? When DNA synthesize the molecules other than itself. Okay, understood? Like the synthesis of RNA in the process of transcription. Again one process, that is the synthesis of protein in the process of translation. This heterocatalytic function, inside that DNA synthesize protein, DNA synthesize RNA. That's why we include it as a heterocatalytic function. Then another is autocatalytic function. Then what is meant by autocatalytic function? In this, DNA synthesize itself. DNA synthesize itself. For example, that is a replication. We form a replica. It is a meiosis or mitosis. In that process, we form a replica. Then process by which DNA replicate itself. And this process, replication process, is that process in which DNA replicate itself. Right? Then in eukaryotic organism, replication of DNA takes place only once. This is a very important point. That is, in eukaryotic organism, replication of DNA takes place only once in the cell cycle. It occurs in the S phase of interphase in the cell cycle. Inside the cell cycle, in S phase of interphase in cell cycle, this is takes place. Then DNA replicates through semi-conservative mode of replication. DNA replicates through semi-conservative mode of replication. The mode of semi-conservative replication was proposed by Watson and Crick. You know that Watson and Crick model. This double helical structure is observed by this Watson and these are the nitrogen bases, phosphorus compound ET, like this. This is a DNA. Who proposed that? That is our Watson and Crick. On the basis of anti-parallel and complementary nature of DNA strand, anti-parallel, this one strand and this is one strand, that is anti-parallel and complementary nature of DNA strands, the process of semi-conservative replication is like this, like is here. And this first point is here, the process of semi-conservation here. First is, that is a activation of nucleotide. First is activation of nucleotide. Activation of nucleotide. What happened here? There are the 4 types of nucleotides of DNA. That is DAMP, then D, that is DGMP, then another DCMP, and last one is DTMP. These are the four types of nucleotides of DNA, that is DAMP. What is meant by DAMP? That is D stand for D stand for DNA. D stand for DNA. Then AMP stands for adenine monophosphate. That is adenine monophosphate. D stand for DNA, and this AMP stand for adenine monophosphate. Then D stand for guanine monophosphate, C adenine, guanine, and thymine. All these four types of nucleotides are present in nucleoplasm. Where is nucleoplasm? If this is the nucleus, if this is the cell, inside and centrally are in the nucleus. Here is the nucleus. This nucleus surrounded by cytoplasm, and inside the nucleus we found nucleoplasm. Understand? Inside the nucleus we found a nucleoplasm. They are activated by ATP in presence of an enzyme, that is phosphorylase. That enzyme, name of the enzyme is phosphorylase. This phosphorylase results in the formation of deoxynucleotide triphosphate, that is here. They are converted to like this. That is here. This DAMP is converted into DATP triphosphate. This guanine converted into triphosphate like this. All those are converted, and this process is known as phosphorylation process. This process is known as phosphorylation process. This DAMP converted into DATP molecule is known as, the process is known as phosphorylation process. Then our second point is, this is about the activation of nucleotides. Then second point is point of origin or interaction point, initiation point. Point of origin or initiation point. This is the second step of DNA replication. Here, what you observe, that in first activation of nucleotide, all those nucleotides present inside the nucleus or DNA molecule, they are activated. Then point of origin, from which point that DNA molecule they get replicated. They form their replica. For that, in this point, they begins at a specific point O. If this is a after this double helical uncoiling, we form this ladder of DNA molecule. This 3 strand, 5 dash, 5 dash, 3 dash, right? Then if this is that point, origin point, that begins at specific point, that is O, that is known as origin, and terminate at a point T. Okay? Then termination. For example, here in this termination point, the origin in flanked by T site and unit of DNA in which replication occurs is called a replication process. This is from here the process is called a replication process. In prokaryotes, there is only one replication. However, in eukaryote there are several in together. 7 replication is occurred. They are in together. Okay. Then our third point is what? Unwinding of DNA molecule. Unwinding of DNA molecule. Now, enzyme DNA helicase operates by breaking a weak hydrogen bond. Inside this, there is presence of hydrogen bonds, weak hydrogen bond. In the vicinity of O strands, this is that O strands of DNA, separates and unwind. Separate. Here, they get separated and unwind. This is winding, and after that they are unwind there. This unwinding is bidirectional and continuous as a Y shape, like this. Continuous like a, this is Y shape. Bidirectional and continuous as a Y shape replication form. Each separating strand acts as a template. Each separating strand acts as a template. This here also and here also. This is called a template. The two separating strands are prevented from the recoiling by SSBP. SSBP. What is this? Single strand binding protein. What is that? Single strand binding protein. And what is the role of this SSBP? The role of SSBP is that when they are unwind and they form ladder, and after that they form this Y-shaped structure. In this Y-shaped structure, when these templates are get formed, then these templates, when they get separate, and after that separation, they are not come together. That's why this SSBP protein, they are control the activity or the prevent these strands or these templates to come too close together. This is the function of this single strand binding protein. Okay. Then this SSBP remain attached to both the separated strand. This SSBP, they are attached with the separated strand so as to facilitate synthesis, that is a formation of a new polynucleotide strand, Then our fourth point is that is replicating form. What are these replicating fork? The point formed due to unwinding. The point, look here, the point formed due to unwinding and separating of two strands appear like Y-shape, Y-shaped fork called replicating fork. Understand? Is that position from there, this strand, they get separated from each other, and that point is called a replicating fork. Okay, and that looking like a Y-shaped structure. Then again, the synthesis of new strand. Next point is, fifth point is synthesis of new strand. Synthesis of new strand. What happened here in synthesis of new strand? The separate strand act as a template. This separate. If this is, these are the separate strand. They act as a template. These are our here. If this is A, T, C, A, G, like this. If this like this only. For example, we are just considering is that. Then this is a leading template. This fork is considering as a template. Right? Then these are the separate strands act as a template for the synthesis of new strand. For the synthesis of what? New strand. Then this new complementary strand began with a small RNA called as RNA primer. This formation of new strand is began with whom? Began with this RNA primer. The RNA primer complementary nucleotide from the surrounding of nucleoplasm. Nucleoplasm, I told you in the that it present inside the nucleus. The nucleotides and the complementary nucleotides on the template stand by forming a hydrogen bond. That is, they form this like A T here, AT or TA, this double bonding structure, or and G triple bond C or C triple bond G. This like this a formation of bonds. They connected by a phosphodiester bond forms a polynucleotide strand. A new complementary strand is always formed by 5' 3' direction. Right? This new strand is formed like this 5'3' direction. Okay, this is a new strand. Then next is leading and lagging strand. Next is leading and lagging strand. The template strand with free 3' end is called leading strand. This is that leading strand. Okay, with free end, 3' end, and this is called as leading template. With free 5', this is 5'. This is free 5' end is called as lagging template. Lagging. This is a lagging template. Like the presented DNA are anti-parallel, new strands are always form 5' to 3' direction. That is 5' to 3' direction. These are the formation of new strand. Then next point is that is a maturation of Okazaki fragment. Then what is maturation of Okazaki fragment? When this RNA primer they are forming, and there are some continuous strands are formed, and that fragments are called as Okazaki fragment. Why we told this as a Okazaki fragment? Because this is the name of the scientist who discovered and who told about these fragments. That's why we named it as a Okazaki fragments. DNA synthesis and lagging templates takes place in the form of a small fragments. Here these are the small fragments of DNA. DNA synthesis is a lagging templates takes place in the form of small fragment called Okazaki fragments, and that derived from the name of the scientist. That Okazaki fragments are joined by the enzyme, that is DNA ligase. Then RNA primers are removed by DNA polymerase and replaced by this DNA sequence with the help of DNA polymerase. The DNA polymerase 1. Then in prokaryote and polymerase in eukaryotes, finally DNA isotopes are formed there. DNA gyrates are formed there. Okay. Then enzyme from double helix to form a daughter DNA nuclei. From this enzyme, due to that presence of topoisomers, the enzymes form a double helical to form a daughter DNA molecule. How the daughter DNA molecule is formed? Here if this is a molecule, this is that molecule. Here what I observed from here what happened? No, sorry. This is the DNA molecule. From here and from here. Is it a DNA molecule? Then from here what happened? Like this structure. From here is observed like this structure. This is a old strand. Then from here there is a formation of new strand like this. The formation of new strand like this. Here also a formation of new strand. Here is formation of new strand takes place. Here we found old strand. These are the old strand. These are the new strand. And this is a daughter. This is a we call it the daughter molecule, daughter DNA molecule. And this one also a daughter. Now from a single molecule, DNA molecule, there is a formation of two daughter molecule. This is that process. That all these points we discussed above, in that process, what happened? There is from the single DNA strand, there is a formation of two daughter molecules. Clear? Our last point is formation of daughter DNA molecule. This is that. Formation of daughter DNA molecule. Then, what is that process? The end of the replication, two daughter DNA molecules are formed. In each daughter DNA, one strand is parental. This strand is parental. Then, and other one is totally newly synthesized. This is newly synthesized. Thus, 50% is contributed by mother DNA. This 50% is contributed by mother DNA. Hence, it is described as a semi-conservative replication. It described as a semi-conservative replication. Why it is called a semi-conservative replication? Because inside there is a 50% from the parental and 50% is a new formation of new molecule. That's why we call it as a semi-conservative. Semi means half. Here from parent and this is a new cell. Okay, this is all about our today's DNA conservation or